So we're just gonna get started here. So hi, thank you for coming to the first Wildly Called event of the term. We're so happy you could join us. My name is Kayla, and together with Alex, Ilsa, and Hannah, and Shuchi, who isn't here, we help to coordinate these seminars. Before we get started, I just want to acknowledge that here in Saskatoon, we are on Treaty 6 territory and the homeland of the Métis. We pay our respects to the First Nations and Métis ancestors of this place and reaffirm our relationship with one another. So I would like to introduce Patrick. He is a PhD. He has a PhD in molecular neurobiology and is currently working as a postdoctoral fellow at the U.S. He obtained his bachelor's degree in environmental management from the Federal Institute for Education, Science, and Technology in Brazil. And afterwards, he started his master's in cellular neurobiology, followed by a doctorate in molecular neurobiology at the Federal Institute of Periera in Brazil. So we're so happy to have you here, Patrick. Just a quick note, questions will be taken after the seminar. You can just raise your hand once it's done to ask your question, or if you're a little shy and anxious, because I get it, we also have pieces of paper that you can write your question down on, and Hannah will come around and collect those after the seminar, and then we can read them out for you. So without further ado, I'll turn it over to you.
Hello, everyone, again. So thank you for inviting me to present my work. And my title is Migration Neuroecology, Neuroecological and Molecular Mechanisms, which is basically a summary of my PhD, which I work especially in molecular biology. Thank you. Oh, I'm sorry. Thank you again. Okay, so let's go. So I first talk about what I worked at before. In my master's degree, I worked with my thesis comparing migratory with non-migratory birds, especially focusing on the cellular biology. We are trying to figure out if the birds with different migratory behavior have difference in the hippocampus. The hippocampus is a region of the brain which have it's known as involved with the migratory traits such as memory and spatial localization, processing of the visual cues such as landing cues, and also smell of the place stopover sites, and etc. So we compared these two birds, which is the plovers, semi-palmated plover and colored plover, which have difference because colored plover is non-migratory birds. It's native from Brazil, and semi-palmated plover, it's a migratory one. So we discovered that the migratory plover have more mature neurons in the hippocampus, immunostained with the NELN immunomarker, and also have a better neurogenesis. Neurogenesis is the capacity of the brain to born new neurons, and also have a greater cell activity in the hippocampus. And however, when we submitted this paper, the reviewer asked us an important question, which was maybe the difference we found, it's due to the fact that it's not the same species. So, for example, if we compare our number of cells in human beings with the dogs, we probably will find a greater number in humans. But the reason is not because our behaviors are modifying this. It's because it's our different species. So the reviewer asked us about this, and in the master's degree, I started working with the genetics, and we did analysis called the phylogenetically independent contrasts, or PIC, where we used the phylogeny of this group together with the variables we are studying, in our case, number of cells and cell activity and neurogenesis. And we demonstrated that the phylogeny doesn't interfere in the difference we are found between both species. suggesting that probably the differences came from the migration differences and not the phylogeny. So yeah, that is a summary of my master's. And after that, in my PhD, I will explain better in the next slides because the main presentation is about it. But it's working with the next generation sequencing applied for the cellular cell expressions in the brain of the sand-palmated sandpipers, and how the migration behavior affects the gene expression and which genes are involved in the migratory behavior in the wintering zones, which is in Brazil. So my main interests are behavior. I worked with behavior in mice, birds, dogs, monkeys, and other species, such as worms. Believe me, yeah, behavior in worms. And I also worked with flow cytometry. It's basically know how the blood cells are affected by any variables such as infection. We worked with LPS in the blood and measure this through flow cytometry. I worked with three-dimensional morphometry, which is basically see how the shape of the cell changes when in a specific condition, so we can measure this. And RNA-seq, which is the new one, and also stereology which is the cellular countings and estimates, and bioinformatics, which is basically the processing of the RNA-seq data. So before talking about the data, we need to talk about migration. So what is a migratory bird? A migratory bird, it's adults that move seasonally to a certain region as a wintering grounds or even for breeding. So as you can see in the video on slide, so the video stopped, okay. This presentation is a little heavy. This is the native areas of the sand-palmated sandpiper. This is in Cold Island here in Canada. And usually in this area, these birds breeding. So, however, when the fall starts and winter is coming, these areas change drastically, and the food availability, it's decreased. So these birds are forced to be moved in the direction of the southern hemisphere to get better conditions. So they fly to the Bay of Fundy, and there they can find like more food, and they can create new energy reserves and store in the form of lipids. After that, they move to Brazil, especially all the coast of the Atlantic Ocean, but Brazil is a place where they go. And here you can see my field work in Brazil. It's a forest of mangroves, and they can find a lot of food there, like insects and, So, okay, so the migration causes a lot of change in the life story of the bird. One of the change is the hormonal change caused by the exercise. And we know that the hormonal change can improve spatial memory and learning. And the exercise also improves the learning in migratory birds. And also change neurogenesis rates between birds that are migrating compared to the birds that are not doing it. So in 2016, we published our first efforts comparing two model species. It's not a model species actually, but it's two sandpipers, sand-palmated sandpiper on the left side and spotted sandpiper on the right side. And these guys have a particular difference during the migration. They come from the same place, Cold Islands, and then On the left side, you can see the spotted sandpiper path. On the right side, you can see the simple-mated sandpiper path. Spotted sandpiper have multiple stopover sites during the flight. These are GPS data. So in the opposite, simple-mated sandpipers flies directly across the ocean in a flight of 3 days without stopping. So we compared, and our hypothesis is this different migratory behavior may affect the brains regarding the number of cells. And yeah, we found that for the mature neurons, we don't have significant differences. Let me move this a little bit. We don't have significant differences here. And however, the hippocampal volume, it's changed drastically in the spotted sandpiper, which is the green one. And probably the behavior of stopping during the flight causes this. Why? Because during the stopover sites, the birds need to remember the correct sites, the amounts of predators and the dangers. And so this stimulus probably causes a greater volume in the hippocampal traits. Okay. So in 2017, we published a paper where we showed that simple-mated sandpiper compared with different experimental groups. Here we have only one species. Birds collected in Canada, the green ones, in the Bay of Fundy. So they are supposed to be migrating because they came from cold islands to the Bay of Fundy, compared to the birds in the wintering zones, which is the orange one. After migration, the number of newborn neurons are greater. So suggesting that the migration also improves the neurogenesis in the simple-mated sandpiper. And the same for the hippocampal volume, showing that hippocampal plasticity during the migratory process and after migratory process, actually. So we have a lot of examples in the literature about how the genetics can affect how the migration can affect the genetic mechanisms. And as an example, this species, it's a nest parasite. It puts the eggs on the nest of other species outside the residence zones. And then after the chicks grow up, they can go to the native zones without being there during their lives. So showing that there is a molecular background behind the migratory process. And also in the black-capped cicadas, the molecular mechanisms that modulates the migrations can be altered, can be changed by the artificial selection. And this suggests that the migrations involves both genetics and behavioral change, behavior interactions. In 2011, a paper was published studying the size of the gene ADCYAP1, where they have a microsatellite region. Microsatellite is just a repeat of nucleotides in tandem in the gene, inside a gene, such as AT. You have a correlation between the microsatellite size of gene size, we can say that, with the migratory distance. So how far the black-capped cicada migrates, bigger is the gene size. So these genes codes a neuropeptide called PACAP, which is important for a lot of migratory processes, such as change in the body temperature, which is important for regulating the metabolisms during migration, inhibition of the feeding behavior, So the bird can't feel starving when he is migrating. And so these genes have a potential to modulate some characteristics from the bird behavior and the body during migration. So the next generation sequencing comes with a good option to study the migratory process in the brain, because allow us such as development of DNA marker with the global genome coverage and analysis of the entire genomes or transcriptomes, discover of the new genes and characterization of the expression, detection of the gene in the whole brain or even in a whole area such as the hippocampus, and detection of the candidate genes which can be involved in those processes. So some studies already done in using next generation sequencing in birds, and such as the variation of the gene discovery and gene expressions in the song nucleus of birds, depending on the season. So depending on the season, the birds uses a specific set of genes to regulate the songs. And also social behavior that is modulated for specific genes in the zebra finch, and a comparison between migration and reproduction. So as you can see, genes regulate everything, and the specific set of genes can change social behavior, reproduction, and other important things in birds. So here is a model. I use it for my PhD, which is a simple-mated sandpiper. It's a small bird, have from 13 to 15 centimeters of size and 20 to 41 grams. On the right side, you can see the migration, the distribution of the species, where in orange, you can see the native zones and the breeding grounds. And in blue or green, I don't know, it's the wintering zones. And especially here, it's the area where I live in Bragança. And the red arrows is the migration, it's the fall migration, when the birds are like escaping from the winter here and going to the south hemisphere. And the green arrows, it's the migration back to Canada. So I prepared here a small timeline showing our experimental groups, which was 2 groups. And here I have birds collected in September, which is the recently arrived set of birds. So they're supposed to just arrived from migration in Brazil. And 7 months later, I collected some other birds, which is the growth of pre-migratory birds. They are preparing to back to Canada and breeding in July, probably. So the main goal of my PhD was analyze the effect of post and pre-migration life story in the simple-mated sandpiper during the wintering by searching for differently expressed genes which can be related for both states. So as methods, we sacrifice the birds using anesthetic called isoflurane. And after that, we perfuse the birds using RNA leader. Perfusions basically is to put a needle on the left side of the bird's heart and pump with a solution called saline solution. After that, you just cut the jugular of the bird and let the blood flow out. So you do this to remove the blood from the brain. And after that, you change from saline to RNA leader, which is the solution will preserve RNAs in the brain. And after collecting the brains, we did the RNA sequence using the ION platform. And this was the the kits we use it to do it, and we sequence the fragments in the iron proton sequencer by Thermo Fisher Scientific. Okay, after sequencing, we go to the computer part, which is the post-sequencing analysis, such as bioinformatic analysis. So after sequencing, you receive a text file. It's actually a text file with like 1 terabyte size, and with the reads, we call reads, it's the fragments of RNA. So during the protocol, you just fragment the RNA of your sample, and you collect them by sequencer reads them and call as reads. So you receive a text file with the reads, and then you need to filter the low quality reads, which I did with the FastKey and Trimomatics, and we assembled the Trinit, the transcriptome using the Trinit software. To identify the transcripts, we used Blast Plus, and I don't know how to pronounce the name of the fish, but whatever. Because it's tricky, right? I can say a bad word trying to talk this, but whatever. So we used both softwares to gene quantification and differential expression analysis, and Blast to go to annotation and functional discovery of the differentially expressed genes. Here, I forgot to use this. So basically, here is a summary of our data. And in blue, we have the distribution of the read lengths. As you can see, most of the reads in the pre-migratory groups are short, between 20, 30 or 25 size basis pair, and the different from the recently derived birds. However, as you can see here on the table, we have an overall alignment rate of 89.12, which means that 89.12 of sequenced reads was used in the assembly. So this is a good result, because you need to usually a minimum of 80% of usage. Here we compared our transcripts with a database that I assembled from the NCBI, which have all taxons available at NCBI. And as expected, our data is similar to birds, 99%. And inside the birds group, it's more similar to Calidris pugnax, which is the closest related species available there. So this is like a quality check of the data. No, okay, it's right. Here, it's a graph called volcano plot, which we have well, I will explain, I will try to explain this one. So in the vertical axis, we have significant values, such as p-values, but it's we call FDR because it's the p-value transformed or corrected for multiple comparisons. So as is in the log, and it's a negative log, higher value means lower. So dots here on the top have a very, very low FDR value. That means that is highly significant. And bottom ones, it's the opposite. In the horizontal axis, we have the fold chains. Fold chains means that how much the gene changed the expression when you compare state A, which is recently arrived birds, versus state B, which is the pre-migratory birds. And we have this dashed gray lines, which is the linear we set. It's the thresholds. So here, from to top, is significant, and here to right, or here left, sorry, and here to right, it's also significant. The blue dots, each dot is a gene, and blue dots are significant only for fold chains, and gray dots is not significant for any one of the analysis. The red ones are genes that are down-regulated. that means that decreased the expression when you go from recently arrived bird to a premigratory bird. The gene decreased the expression. And the opposite for the green ones, which is genes that increase the expression. So these genes, it's higher when the birds arrive and birds is almost going back, and these ones are lower. Here I put the labels for some genes, but I can't put for all of them because it's more than 600 genes. So it's not possible to see if all the labels are on here. I choose something around topic 20 identified genes. And we found 259 down-regulated genes and 357 up-regulated genes. Okay, so here we have a sample correlation matrix on the left side and expression matrix, a heat map showing the expression in the right side. Okay, so here in the sample correlation matrix, it's just a matrix where we are doing correlations between samples. Here we have samples ID, and for blue for the pre-migration group, and orange for recently arrived birds. And as you can see here, there is a cladogram, which is the relations between the samples. It's important to say that the relations between samples is done by their expression of the genes. It's not like just because I said to the program or to the software that the bird belongs to a specific group. It's not grouped by this, it's grouped by the expression. So this confirms the initial hypothesis that we have, in fact, differential behavior of the gene expression comparing recently arrived birds versus pre-migratory birds. Okay, so here we have the sample labels. It's just a code. And for red values, we have a negative correlation. That means they are not correlated. And the green values is positive correlation. So as you can see here on the top, all birds from the same group are correlated together in the same group, except for this one, which is a bit different from that ones, for the other ones, but not enough to be in another side. Okay, that's it. And we have here negative correlations and positive correlations. Here we have the expression heat map, where we have on the top the same cladogram as, oh, I'm sorry. How can I, oh, okay. Sorry about it. So we have the same cladogram showing the relations between the samples. And then we have a new one, which is the relations between each gene. Here we have more than 600 lines, and each line represents the gene. Green ones is the high expression, and the red ones is low expression, which means down-regulated genes and up-regulated genes. So here we have genes that in the first batch of birds have high expression, and the expression downs to the another group. Here we have some gene labels, and we have a batch of 2 different clusters of genes. As you can see, the cluster of genes have opposite expression. We have genes that have high value to down, and down, up. And here's just a zoom in just to show like closer, and also the same for the bottom. And here we have the first subcluster of genes and the second one. And if we look about the expression of these clusters, we can see that exactly better summarized, I think, that for the first subcluster, we have a low expression and the expression go high, and the opposite for the second one, the expression is high and drops. And regarding the identification of the genes, it's in Portuguese, sorry. So we didn't identify the 11% of the 600 genes, and 41% it belongs to others, which is any kind of function outside of these categories, and 39% is ribosomal genes, and 80% belongs to mitochondrial process. So just recap the experimental groups. We have recently arrived birds and pre-migratory birds. And just as a recap, we did the next generation sequence of the data, of the brain data, and we used the train to assembly. We did the differential expression analysis to test the hypothesis if we have differential expression between genes. And we found 259 upregulated genes and 357 downregulated genes. And also we did the ontology to identify those genes. Okay, so I highlighted here two genes which I used on my discussion, which is the GABA receptor B on the right side here. The GABA receptor is upregulated, which means that increases the value of the expression. And on the left side, I marked the ARHGAF9, which is also regulates the GABA receptors, but it's the GABA A receptor. And here is the GABA B receptor. And I will talk more about those genes. So as you can see, we have a lot of genes discovered to be evolved in the different stages of the wintering of these birds. And I will not put everyone, don't worry about it. I will talk about only of these two ones. So for memory proposed, hippocampal circuits, as we know, it's important for learning because hippocampus is an integrative area of the brain that receives the information about everything the bird uses for migration. And it's important for the social behavior between the birds. I put this video showing the V formation of the geese, which is a strategy to have better aerodynamics during the flight. So this saves energy. Okay, so in 2022, we published a paper analyzing the parvalbuminergic neurons and the hippocampus of spotted sandpipers, and we found a similar difference in the numbers as we found in the gene expression to sand-palmated sandpipers. And if you can do extrapolation about the parvalbuminergic neurons, we can suggest the hypothesis that the GABAergic connections or changes in the brains are essential for migration back, because otherwise they will not change during migration states. So regarding GABA receptor, we know that experience-induced plasticity in the parvalbuminergic neuron cells regulates the learning, and this is already published. And we know also that memory acquisition and consolidation is also associated with the neurogenesis of these kinds of neurons. And we also know that learning and memory are modulated by hippocampal GABAergic neurons, and especially for GABA B receptor. And we also know that synaptic plasticity of inhibitory neurons provides long-term change in the hippocampal circuits and can be a key component for the memory formation. So hippocampal circuits, the hippocampal is a part of the activity network used to integrate the multisensory cues, as I said before, and the migration move in the hippocampal formation, it's used to correct the direction during the flight, and as well the recognition of the stop move site, just as the spotted sandpiper can do. And and talking about the second gene, which is the down-regulated gene, it's also called the guanine nucleotide exchange factor, and also regulates the GABA A receptor. So, however, despite both genes I've highlighted here are involved to GABA receptor, we found a weird behavior in their expression, which is basically that they are opposite. Why two genes that regulate the same thing have different expression patterns, are down regulated or up regulated in opposite. So the GABA A receptor is a channel that mediates rapid inhibitory signals and through the postsynaptic hyperpolarization. And GABA B receptor produces slow and prolonged inhibitory signals. So this is the basic difference between the both receptors. So some questions rises about this data, which is what's the functional role of this contrasting different gene expression for both GABA E and GABA B genes between these experimental groups. We separated our samples for 7 to 8 months, which was the winter period, and this brings our experimental groups close to the breeding time, because when they arrived in Brazil, they are close to the breeding under North America. And when they are almost going back, they are so going to breed. So the proximity of the breeding may affect the data. Okay, so we can also investigate how the GABA receptors are affected by sexual hormones, because we didn't measure that. So this may affect because they are close to the breeding, and we don't know if they can affect the GABA receptor. And yeah, despite the different expression in different GABA receptors, it's just a small part. As you can see, I talked a lot. Most of you are tired to hear me talk. So we can discuss all 600 genes in the sample presentation. And we have a lot of possibilities. This is why I told that next generation sequencing is like fashion, because everyone wants to do. And when you run next generation sequencing, you receive a bunch of data and you will not finish the process until you die. Probably your next generations will work in the same data set. So it's like you use the funds and have a lot of to do. So we also didn't distinguish between the nervous areas in the central nervous system, use the entire telencephalon, because it's impossible to separate the hippocampus of the bird brain unless you have a special equipment to slice on thick sections and isolate it. And the top area is also very unknown, such as the hyperpallion apicale, which is the difference. You can't say the difference between the rest of telencephalon and the hippocampal structures. And that's it. I want to thank you again to invite me, and sorry to speak too much. And I would like to thank you to McGill, which I was another institution that also gave funds for my PhD, and all the Brazilian institutions here. And also I left here this QR code, which is a YouTube documentary showing how is our field work in Brazil. So you will see me 10 years young in this video. So, so yeah, you can watch later because we don't have too much time to do it. Thank you very much.
Quick question, Patrick. Is there any factor you know that was